Hello students, welcome back to the biology class. Today we are going to learn about the Mendel's second law of inheritance, and this second law is the law of segregation. Okay, and this law of segregation is also known as the law of purity. Okay, and this law is universally accepted law of Mendel. Okay, so what is the statement of the law of segregation? Okay, so it is when a pair of contrasting factors come together in hybrid condition, these factors do not blend or mix up and separate at the time of gamete formation. Okay, so what is contrasting character? We have already learned about this, the different types of character and contrasting character. Like height of plant, in height of plant there are two contrasting character: tall height, short height. Okay, then the factor of this tall height and short height come together in one individual, and this individual is known as hybrid. Okay, let's see. This is a tall plant, and this tall plant having two different alleles, capital T as well as small t. So it is a hybrid, capital T for tall and small t for short. But the character of this plant is tall due to dominance effect of the capital T. Capital T dominant over the small t, so the height of plant will be tall. But this is hybrid plant. Okay, so in hybrid plant both different allele or different contrasting factor come together in this organism, but in this organism these both alleles do not mix up with each other. They simply just remain with each other, and they get separate at the time of gamete formation. When this organism produce gametes, it produce two different gametes. Okay, one 50% gametes having capital T and 50% having small t. It means both the allele get separate and pass into different gametes. Okay, they do not mix up and they separate out and form a different gamete. Here also second parent also produce two different types of gametes because it is heterozygous and produce capital T as well as small t. Okay, and then during it produce a different type of progeny. Okay, so we can see it Punnett square, and we can form a different possibility of genotype of the offspring. Okay, so we already know capital T small t. Here we have to write the gametes, and here these are the possibility of the genotype of offspring. So the these alleles separate out and mix to form capital T. It means tall and homozygous dominant. Okay, here capital T small t. Capital T comes from here and small t comes from here. So it's capital T small t. It's a heterozygous and tall plant. Okay, here also capital T comes from here and small t. Capital T small t. It's a heterozygous and tall plant. Again, this small t. Okay, small t comes here and it produces a short plant. Okay, so what is the phenotype of small t? It's a short or dwarf plant. Okay, so we can see that small t progeny, it's this character is not of the parent. Here both the parents are tall, but the progeny will be short due to segregation of allele. Okay, and this is the law of segregation and purity. Okay. Hello students, now see the next topic is a taste cross. Now what is taste cross? Taste cross is cross done between two plants. One is F2 dominant plant. Here F2 dominant plant cross with F2 dominant plant with its recessive parent. Okay, the cross done between the F2 dominant plant with its recessive parent is called taste cross. Okay, now what is F2 dominant plant? Okay, so F2 dominant plant, if we take a character like a tall and short, then the dominant character is tall. Okay, so the F2 dominant plant is tall and recessive character is short. Okay, now this cross is called taste cross. Okay, now why Mendel had done a taste cross? Okay, what is the importance of taste cross? So it is done to find the unknown genotype of F2 dominant plant. Okay, now what is unknown genotype? Okay, now we can say the plant height, and we can see that this plant is tall and this plant is short, but we cannot tell the exact genotype of the tall plant. Okay, now let me see the example. If we see the short plant, then we can see that the genotype is small t. Okay, so now here the character tall and short. Now the genotype of short plant is, we can see that small t because recessive character, short is a recessive character, and recessive character having two allele small t we know. But now what is the genotype of tall plant? Okay, one condition is One is capital T, it is a tall. Okay, tall plant genotype is capital T, and another is capital T, small t. It's also a tall plant due to dominant effect of capital T. So the tall plant having two genotype, capital T, small t. So we cannot say that the tall plant having which genotype by seeing. Okay, so this technique, test cross, is the technique to find out the exact genotype of a tall plant. Okay, for that Mendel had done a cross between its recessive parent. Okay, so we have to take a two case, one for capital T and one for capital T, small t. Okay, let's see the cross. Okay, so first of all, in case 1, we take a tall plant having genotype capital T. Right now we are not knowing the genotype of this plant. Okay, it's unknown. Okay, now we have crossed a capital T plant with its recessive parent small t. Okay, now the gametes of this plant is capital T and gametes of this plant is small t, and the progeny having genotype is capital T, small t, but the phenotype of all the progeny is tall due to dominant effect of capital T. So capital T, small t, it's a tall plant. Okay, so if we get a 100% tall plant in progeny, then the genotype of unknown dominant plant will be capital T. Okay, got. Now next case 2, parent having the genotype capital T, small t. Right now we are not knowing the genotype of this plant, tall plant, capital T, small t. Okay, now if we done cross with the recessive parent, the gamete of this plant is capital T, small t, and these plants gametes are small t. And if we form a Punnett square and find the result, probability and of genotype, then we get a capital T. We have to write the gametes here, capital T, small t of this plant, small t of this plant. Then we get the probability, capital T, small t here, it's a tall. Here capital T, small t, it's also tall. Now here small t, it means short. Here also small t, and it is short. Okay, so we get the 50%. These two are tall plant, so we get the 50% tall plant, and these two are short plant, 50% short plant. So if we get this result, 50% tall and 50% short, then we can say the genotype of F2 dominant is capital T, small t. Okay, so again, if we get result 100% tall plant, then the genotype is capital T, and if we get 50% tall and 50% short, then the genotype is capital T, small t. It means heterozygous. Stop.